In this screencast, we'll talk about amino acids, their ionic forms, synthesis, and polypeptides. So our first key concept is we want to understand the functional features of an amino acid. So the amino acid structure is the following. You have the carboxylic acid moiety, our alpha carbon, which we defined as the carbon next to a carbonyl, has the amine, and then so this is the amino acid part, and then this R group is the side chain. So most amino acids are of the S stereochemistry, so the naturally occurring essential amino acids are S. So this L refers to the Fisher projection, and basically what we're doing from this convention to the Fisher projection is we're putting our eye in the plane of the paper, and then we're looking at down the carbon backbone. So if your eye is looking here, this carbon, which is the highest oxidized carbon, is at the top. This carbon here, which is the alpha carbon, that's a stereogenic carbon, meaning it has 4 different substituents. It's represented by this horizontal line here. So on the left is the amino group, then on the right, which is going away from you, is the H, and then the R group, which is the side chain, is down. So in the Fisher projection, this is defined as the L amino acid, so the capital L. The Coningal prelog is this, if you determine that, that's the S. The side chain can be polar or nonpolar, and it can have acidic or basic functional groups. So proteins are biopolymers with amino acid monomers bonded via the amide linkage. So this is an example of a tetrapeptide. And so let's just go through the structure of this small polymer. It's basically consisting of different amino acids. So the way we interpret this on the left-hand side is called the N-terminal residue. And then as we read it from left to right, we end with the C-terminal residue. And so what you're going to want to interpret as you're reading this is you find your amine group bonded to the alpha carbon. Here's your carbonyl, and now you have this amide bond. So this is made by the condensation of this amino acid, which is cysteine, bonded to this next one, which is serine, and we get those names based on what the side chain is. So we're looking at the alpha carbon, what the R group side chain is. This first one is cysteine, this next one is serine. So these amino acids are bonded via this amide bond, otherwise known as a peptide bond. So if this is the acid, it's been condensed now with the amine of a second amino acid. We're reading along here. Here's the second peptide bond, and we look at what amino acid this third one is here. We look at this side chain, that's valine. We're coming along, there's another peptide bond. Here's the alpha carbon, and then this side chain is phenylalanine. So this is the chemical structure of that tetrapeptide drawn out. It will be abbreviated using a 3-letter abbreviation of the different amino acids. So CYS is cysteine, SER is serine, VAL is valine, PHE is phenylalanine. So cis, ser, val, phe. So when you're looking at this, your mind should be envisioning this right here. These letters can be further abbreviated from the 3 letters to 1 letter, so CSVF. So again, the same thing. When you're looking at this, you think of this and this. So when amino acids bond to each other via the peptide bond, you have to consider that there's amide resonance. And so that has implications in the geometrical isomerism about the peptide bond. So if we consider R prime coming off one amino acid. Here's the peptide bond, and we have our other amino acid over here. So we're interested in this, the geometry about this bond, because this lone pair can be delocalized through resonance onto the oxygen, as drawn down here below. So now when you do that, you have this CN double bond with plus charge on nitrogen, negative charge on oxygen. And so what this has the implications of is now you have a geometrical isomer. And determine whether it's Z or E, you look at the substituents on each side. So if this is carbon versus oxygen, oxygen gets priority as 1. Then on the nitrogen, you're going to have carbon versus hydrogen, so this gets priority 1. 1 and 1 are on the same face of that double bond, so this is Z. So keep in mind, based upon having 1, 2, 3 peptide bonds, each of these can be either E or Z. So of a protein, you have 4 different types of structure that you need to understand. The primary structure is the exact sequence of the amino acids. The secondary structure is the ordered structure due to hydrogen bonding, and so that's depicted here in sort of a cartoonish form. So really what we're looking at is a very long amino acid chain, and so this just indicates that it wraps around itself, and then it has these intramolecular hydrogen bonds. So this would be a hydrogen bond donor, this would be the acceptor, this is the donor, this is the acceptor. So that attraction gives it secondary structure. The tertiary structure is the complete 3D conformation of this, and you'll learn more about that in biochemistry. And so a quaternary structure is the association of 2 or more peptide chains. So you should be familiar with this terminology if you go on to the biochemistry sequence. So our key concept 2, understand the ionic forms of peptides. So amino acids typically bear a charge due to the amino group and the acid group. So depending on what the pH of the medium is, you can have cationic, neutral, or anionic forms. So let's look at what those can be. When you're at a pH of 0, and again, you're in the presence of extra acid, basically the amine group is going to be protonated. That's called an aminium ion. And so if you look at this, you're going to have a cationic form. You have an overall plus charge on the amino acid. If we're going to the right, in which we're adding base, eventually we're going to hit the pKa value where this acid gets deprotonated by the base. So remember, carboxylic acid has a pKa of about 4 or 5. So once we reach that threshold, this gets deprotonated. Now this is the carboxylate. This is still protonated under those conditions. This is called a dipolar ion, or a Zwitter ion. Zwitter ion just means it's an internal salt. The plus and the minus are within the same molecule. And this is the neutral form. As we continue to add base, hydroxide, eventually we're going to reach a pH where it's able to deprotonate this pKa value, which is about 10 to 12. So once we get that sufficient concentration to deprotonate this, we're now in the anionic form, where the amine is neutral and the carboxylate's negative. So I'm just showing you broadly from pH 0 to 14 what these different forms can be. And that's going to change depending on if the side chain has an acidic or basic functional group as well. So moving on to some synthesis reactions of amino acids. The first one is the Gabriel malonic acid synthesis, and this is a combination of the Gabriel amine synthesis with the malonic acid synthesis we learned in Module 3. So if we start with potassium thalamide, This is a source of nitrogen that can only undergo monoalkylation. The substrate we're going to use is diethyl bromomalonate. So diethyl, that's the diester, the bromo, and then these 3 carbons, that's the malonate. DMF is the solvent, dimethylformamide, so this is an SN2 reaction. The nitrogen will attack here, displacing the bromine. You now end up with this alpha amino malonate, so it's protected. We have an acidic hydrogen. Remember, we're sandwiched between 2 ester groups, so we have a fairly acidic proton. We're going to treat with sodium ethoxide and ethanol. And then this is going to be our alkylating agent. So after we generate the enolate here, it will attack this carbon, chlorine will leave. So we've now done a substitution of that hydrogen for this side chain here. The next step, we're going to saponify off the ester groups to get the dicarboxylate. We then treat with acid and heat to do a decarboxylation. What that's also going to do is, in this process, is cleave off this phthalimide protecting group and hydrolyze off the amide at the end. So we're removing this protecting group as this byproduct. CO2 is lost from the decarboxylation, and we end up with our amino acid. So another amino acid synthesis is the Strecker amino acid synthesis. If we start with an aldehyde, we treat with ammonia and hydrogen cyanide. The ammonia is going to form an imine. The hydrogen cyanide will then add to that imine. So this first step, we're generating an alpha aminonitrile. So again, this is sort of like a reductive amination in a sense, but we're adding cyanide. So the alpha aminonitrile can then be hydrolyzed with acid and heat. So in this case, we end up with the alpha amino acid in the racemic form, and this would be tyrosine based on the side chain. So here's what happens. The ammonia reacts with the aldehyde to give you the imine. The imine is protonated by the hydrogen cyanide to give you this iminium ion, which is then attacked by the cyanide, and you end up with the alpha aminonitrile. So this has been a short screencast on amino acids and their incorporation into tetrapeptides, a brief introduction into protein structure, and 2 methods to synthesize amino acids in the racemic form.